The broadcast is now starting. All attendees are in listen-only mode.
Welcome everyone to the sixth and final webinar in our series on alternative approaches for acute inhalation toxicity testing. My name is Amy Klippinger. I'm the Associate Director of the PETA International Science Consortium, and I will be co-moderating today's webinar with Dr. David Allen, who works as a contractor supporting the US NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, also known as NICETM. I'd like to thank you all for joining today's webinar, and before I introduce our speakers, I'll run quickly through a few logistics. The slides and a recording of today's webinar will be posted online at the first link on your screen within a few days. The previous 5 webinar recordings are posted there as well. Everyone is muted now. If you have a technical question for me anytime during the presentation, you can use the question box, which is located in the GoToWebinar panel on the right-hand side of your screen. We'll hold all questions for the speakers until the end of both presentations. If you have a question for one of our speakers, you can type it in that same GoToWebinar question box, and Dave will ask your questions to our speakers during the Q&A period at the end. But please feel free to send in your questions anytime during the webinar. The earlier, the better. So our first speaker today is Dr. Kelly Berube. Kelly is the Director of the Lung and Particle Research Group at the School of Biosciences at Cardiff University in the UK. She has a background in electron microscopy and lung toxicology and has built an international reputation in the field of air pollution and human health, and holds numerous appointments in the US and UK on funding bodies, advisory councils, professional societies, and journal editorial boards that focus on environmental health. Her research focuses on the determination of intelligent biomarkers of exposure and harm in the respiratory system, with a particular interest in understanding how pollutants compromise lung biochemistry and alter gene expression and protein expression to drive disease mechanism. Kelly?
Well, thank you for that introduction, Amy. And I'd like to begin by thanking yourself and PETA International for giving myself and my colleague, Zoe Prithik, the chance to showcase our lung models today. So I'm going to be telling you about the normal human bronchial epithelial model and the metabolome model, and we use these for inhalation toxicology applications, but they're very versatile and they rely on using human primary tissues. Okay, so the reason for our research and why we built these models is because of the burden of lung disease. So myself and Zoe are respiratory toxicologists, so I want to set the scene behind our models. For example, globally, mortality for respiratory diseases ranks #2 on the mortality list. And in the United Kingdom and Western Europe, it's #1. And indeed, in the United Kingdom, 1 in 4 people die from a respiratory disease. And the FDA has said recently that there's been no innovation in the last 25 years. And they believe the fact that we're working with animals and that we need to move to human tissues so that we can get some basically useful results, some human data, generate some human endpoint data. And another problem, too, is that air pollution has time before they die. So you can actually lose up to 5 years of your life depending on where you live. Die early, that is. The WHO says in the next 10 years we're going to have 30% increase again in CRDs, that's chronic respiratory diseases. And then finally by 2020, which isn't that long, about 12 million and of course with a lot of people like yourself believe that The reason why we have no innovations and we're stuck with this problem is because of traditional animal testing. And of course, if you're familiar in this field, with inhalation, we use a lot of rodent models. So we use hamsters and mice and guinea pigs, etc. But the problem is that their physiology and anatomy is so different from ours that you really cannot get a good extrapolation. Typically, before the advent of human tissues, we would take the rodents and then we would instill particles into them, say air pollution, and they'd either be fed nose only or whole body exposure, so inhalation or instillation. Then we would just leave them there for acute or chronic times. Then we would euthanize them. We would rinse out their lungs, look for any type of inflammatory mediators. Then we would look at the histology, and then we would try to come to identifying some biomarkers to say, if this happens in the rat, this is going to happen in man. And it just doesn't work. So this kind of led to the advent of human tissue banks and in vitro models, and this is where Zoe and I started off about 10 years ago. We were able to start buying tissues, so we moved away from working with the animals, and we started buying tissues. And we started with Lonza, and we buy their normal human bronchial epithelial cells. They isolate these now from live donors or even post-mortem donors, up to 3 days post-mortem, so that's called medical waste tissues. And what we do then is we get these isolated for us. We remove the basal epithelial cells, so these are stem cells, and then we devise a 3D formula. We have a know-how document on that, so you can buy a license to generate it yourself. Now this differentiates the cells, and the basal cells go right into about 7 different cell types. So the 7 cell types that you find in the bronchial epithelium. This is grown in the air-liquid interface, just how we breathe. And you can get ciliated cells, goblet cells, they secrete mucin. Clara cells, they secrete oxidants when you have an irritation in the lung. Intermediate cells and basal. So the basal cells will turn into the intermediates, and then the intermediates sit there. And if you have a Clara, goblet, or ciliated cell damage, which means you're causing a hole in that area, the intermediate cell will turn into one of those and plug that hole. So it's a very tiny little system. It's only 5 millimeters in diameter. And working with Millipore, we've come up with a system that works quite well using a microporous membrane. So you don't have to have fibroblasts down there or collagen. You can work right off this membrane. And then from there, after the experiment, you can take the tissues and do histology. You can do toxicogenomics, proteomics, take the apical wash and see if there's any proteins or inflammatory mediators. You can look at the basal media through HPLC and see if you have any metabolism going on. Okay, so that's the microlung model. Right, so why are we using that model right now? Why are we going to all this trouble to characterize it? Well, it's pseudostratified, like you find in vivo. You get the 7 different cell types, but they can go layers up to 5 to 7 layers. So if you're working with submerged cell culture, then you'd only get one type of cell, a monolayer, and you, one cell type, just a monolayer. The other reason that we use these NHBE cells is that you get tight junctions and adherent junctions. You also get desmosomes and hemidesmosomes, cilia and microvilli. They secrete cytokines and mucin, and they're non-cancerous, or there's no transfected genes. Right, so basically we've created a mucociliary phenotype, just like you find in the human situation in the lung. So this is the membrane here. These are light microscope pictures, toluidine blue semi-thin sections, basal cells down here. You have the goblet cell. You can see the mucin in there. You can see the long cilia. They're about 10 microns in length, a micron in diameter, just like you find in vivo. Ciliated cells, the Clara cells here, and the intermediate cells. And if you look at SEM, here you see a bit of irritation, and the goblet cell's swollen up, so it's full of mucin. And that mucin will then be released, and the cilia beat back and forth, and they spread the mucin across the whole surface there. And it's like slide paper, so you can trap debris that you inhale and then try to flush it out. All right, and the other interesting thing about our cell culture too is that you get basal bodies and you get real cilia. So what I mean by that is that when you do the transmission electron microscopy, you'll have the axoneme, the 9+2 organization there. Now, when you work in the submerged cultures, you might get little stubby cilia. They're really kind of like microvilli, and they don't have this 9+2 arrangement. So we know we're getting true cilia here. All right, for secretions, well, you can see the mucin here, the glycocalyx, and in vivo, just the tips stick out, and then you have all of the mucin inside there to trap, to act as flypaper, And that's exactly what's happening in vitro as well. They beat synchronously, and if they're in your body, they beat up towards your mouth so that you would remove them through the system by spitting, swallowing, or sneezing. In addition to, with submerged cultures, you won't find this, but you also won't find your tight junctions, or if they do, they're weak. We have lovely tight junctions, adherence junctions, desmosomes, and I don't have an image, but we have hemidesmosomes as well. We see ZO1 in a clothing forming at days 1, and then by day 33, we see all of the tight junctions there. Now, some of the submerged cultures don't even form the tight junctions to this level, so they're a bit leaky. All right, now the system we use is really simple. It's a 3-step process. After you grow these cells, it takes about 2 weeks to grow. Then you have 4 weeks to grow and 2 weeks to use them. You put your compound on, and that's either going to be, it could be a liquid, it could be nebulized powder. Just deliver it onto the surface. You keep it for the amount of time that you're interested in, say acute. Right, then you have to wash it off with PBS, and then you can take that PBS wash, and that can be taken off, and you can do all kinds of conventional tox assays. You can take the tissues off, and again, the basal media, and do everything that you can do right now with normal tissues. Right, so and the important tool for us is called the endome chamber. So we measure tear. So here's the E-bone, here's the chamber, and what we're looking for is electrical current traveling through the apical and basal part of the cell, and that tells us if we have a tight barrier. If it's a tight barrier, it's a healthy culture. If there's a rupture in the barrier, then we know there's been injury. So you put your little insert inside there, and you do the measurement. So high resistance, high ohms, 2,000 ohms, you have a healthy barrier. At low resistance, you'll have holes here. That suggests that the tight junctions have ruptured or cells have actually sloughed off the membrane, and you know you've had some injury here. So that's the principal tool that we use along with conventional toxicology. All right, and here's some example data here. This was 3 different donors to show you that there's very little variability. From day 1 to about day 15, we have this rapid growth, and some people use their cultures from about day 18 onwards, and we wouldn't recommend that because it takes day 21 to get the cilia to grow the full length. So we start using about day 24, and then we can work all the way up to day 36. So it gives you a nice window to do acute, repeat, or chronic studies. Then by about day 36, the culture demises. So it's a 2-week window. All right, so I'm going to show you some examples from Zoe's PhD. So we're looking at LPS here. So she treated the cultures, LPS, and the first thing you see happens is that the tear bottoms out. So that means the tight junctions have become leaky, and the cell doesn't like the LPS, so it's an irritant. And here is the ATP now. That's ATP assay cell viability. You can see the cells are having a bit of hormesis there, but then eventually they can't keep up with it, and they fall down. So you always want the tear and the ATP to be doing the same thing. Here, obviously, we're having a toxic reaction. If you have a look at the histology, it doesn't look like much here in the upper layers and middle. You can see a little damage for TD50 here, TD5. You're having spaces and cytolysis, I think they call it, vacuoles between the basal bodies. But if you look at the SEM, the surface looks okay. So that's why we recommend that you do SEM, TEM, and LM. Because right here, if you look at the surface, I think it's fine. But down here, there's some damage. There's obvious damage here at a toxic dose 20% with LPS. It's throughout the whole system. And then here you see the ciliated cells actually sloughing off and balling up, suggesting apoptosis. There's some necrosis in here. So you get the same type of reactions that you see in vivo in the in vitro situation with this model. Okay, so now we did a little work on cytokines to see if we get any response. So the red suggests that we have severe response, and the blue is a mild response. So this is for the TD5. I'm going to show you a matrix in a minute, so I just want to quickly show you this data. Then we did it for 4 hours. That's the striped red and blue. And again, severe and mild with the colors. But if you take all this information and put it in a matrix, we could learn a lot about what happened in those cell cultures. So at 24 hours for the TD5, we had a severe reaction, nothing at 4 hours, and then at 24, that reaction is still persisting. So that suggests that we have mediators of injury happening here with that cytokine. Then you come down here, TD5, we're having a low level of injury, but by 4 hours and 24 hours, it's persisting, suggesting that you're able to discern between low and severe toxicity here. If you come down to panel B, Okay, we have a severe toxicity here at CD20 for 4 hours, persisting at 24. So that suggests severe injury. And finally, this is a nice one down here, 5. Nothing happening at 24 hours for TD5 and 20. This suggests here at the acute level that we're looking at early indicators of injury. So that's a nice way to use cytokines as a biomarker in the system. And this was an ELISA system as well for the cytokines. It's inexpensive, quick. Okay, so we worked with AstraZeneca. They funded Zoe's PhD. They asked her to look at about 25 compounds, AZ1 to 25. So we were asked to determine if they were irritants or not. And we went into this blind. We used ATP, TIR, and Bradford. And then we predicted AZ14 was not irritant, Z9 was a possible, and 28 was an irritant. We then had to prove this with histology, and we did it here. AZ14, no irritant, it's beautiful. The membrane, 5-7 layers, all the cell types are there with cilia. Now we have mild injury, which we predicted, and now you can start seeing vacuoles. And then the irritant, complete dedifferentiation of the whole system. All we have left here are basal cells, and that's it. And most of the layers, 5 layers, are sloughed off. And then just an added bonus here, I show you AZ1. They said that in vivo, that was not an irritant, but when we did our work in histology, we showed that was an irritant. So in this example here, they did an animal study at the same time that we did the in vitro study, and we came out with 77% correlation, which now suggests that they should be pre-screening with our system, and maybe in the future not even have to go into animals or just do a final proof of concept. So that was quite exciting correlation. Right, the last thing we did, and I can't tell you too much because we're still putting the IP in place and getting our patents, so we haven't been able to publish, is we've got this metabolome now. So what we've done is we've taken donor-matched hepatocytes. So we've taken liver cells from the people where we got the epithelial cells from. We grow the hepatocytes in the well where you keep the food. Then we put so the hepatocytes mature in about 5-7 days, not less than that. Then we grow the NHBs on top. They share a co-culture media which we designed, and they can go for about 10 days. And what you can do is test whether the parent compound is toxic once it's been biotransformed. Example here with HPLC. Okay, we looked at detection of acetaminophen after 24 hours exposure to phenacetin. So here we have nothing. So hepalone, of course, you're going to see biotransformation. Maybe a little with the NHBE, so some inert transformation. And then when you put the two together, co-culture, we had the biotransformation and we got the correct product out. Another example, and we went through the whole classic panels, about 12 different substrates you had to work with the liver to prove that you had biotransformation. Right, so we looked at detection of dextromorphin after incubation with dextromorphin. It's hard to say these words, sorry. So again, we saw the transformation here of the liver, a bit of innate on its own, but when you put them together, we definitely got some biotransformation and we got the product that we were expecting. So that's the metabolome there. That's all I can say about that right now until we can do some publishing. So in conclusions, really, as inhalation toxicity, you saw there's a huge problem that we're facing right now, and they're saying by 2030 that respiratory disease, chronic respiratory disease, will be the biggest killer on the planet by 2030. So we have some problems to deal with. Using animals, we haven't been able to get understanding that we need so that we can come up with some clinical and therapeutic mitigations. So now we're using these pathophysiological models, the metabolome and the microlung, and we're starting to get useful answers, suggesting that we can move away. And Zoe and I are interested in complete replacement. So human tissue-based models, we believe, are the way to go. In terms of all the work we've done, we've characterized the model to death. We know the ratio in the human body of ciliated cells, the goblet cells, etc., all the secretions, everything that's going on. So the model is very well characterized, and we feel that it can now step in and be a viable alternative to using the rodent model. You have a good window to do acute, chronic, and repeat. Remember, primary cell cultures don't last that long, so a 2-week window is very good. And it's very cost effective. One donor cryo vial gives you 500,000 cells, and we can get 350 of these little microlungs to work with, and probably for less than 2,500 pounds, and that includes all the consumables, the media, the bullet kits, etc. All right. In terms of the metabolome, it opens new avenues for us. We can start looking at multi-organ toxicity from just using a filter well bioreactor. And these things are great. You can buy them right off the shelf. They're easy to use. My husband's a geologist, and he's able to carry out this work. You don't need specialist training, and they are affordable, high throughput work, which is nice. And then finally, I think, even though we've built these for inhalation toxicology, there's applications for testing pesticides, for testing perfumes. We've done work for Unilever, testing hairsprays, food products, etc. So it just doesn't have to be limited to working on air pollution. And again, our remit really is we're looking for complete replacement. Okay, thank you. Any questions? I think I did that in 20 minutes. Hello?
So hi, Kelly. This is Dave Allen, co-moderator of the webinar. We do have a couple of questions, but I think we're going to hold those to the end of the webinar.
Sure.
If you don't mind.
Okay. I'll go mute now.
Okay, I think Dr. Clippinger is having some telephone issues, but I'll go ahead and introduce Dr. Huh. Our second speaker today is Dr. Dan Huh. Dan is an assistant professor and Will Family Term Endowed Chair in the Department of Biochemistry at the University of Pennsylvania. He is a pioneer of organ-on-a-chip technology, and his research group at Penn focuses on developing micro-engineered models of human anatomy and physiology for a wide variety of biomedical applications. Dan has won many honors, including the John J. Ryan Medal from the Royal College of Surgeons in Ireland, Design of the Year Award from London Design Museum, NIH Director's New Innovator Award, the Analytical Chemistry Young Innovator Award. He's been a TEDx Fellow, Best Publication Award, and Best Postdoctoral Award from the Society of Toxicology, and finally the Weiss Technology Development Fellowship from Harvard, and many others. I'm very pleased to have Dr. Huh today to talk about microengineered physiological biomimicry, human organs on chips. Take it away, Dr. Huh.
All right, thank you. Thank you for the introduction, and I'd like to start by thanking the organizers for the opportunity to speak at this webinar. So by way of quick introduction, let me start by telling you a little bit about what kind of research we do in my laboratory. So research in my lab basically revolves around developing these microfabricated devices that we use to grow human cells and also mimic the complex structure and environment of human organs and human body in ways that have not been possible using traditional cell culture models and techniques. And the idea is to use these systems as microengineered models of human organs, to be more exact, functional units of human organs for a variety of applications for drug testing, toxicology screening, environmental monitoring, mechanistic disease studies, and so on. So we call these devices or systems human organs on a chip. I'm sure you've heard of it. So what I'd like to do today is to introduce this technology to you guys and help you understand what's possible with this technology using a couple of human organ on a chip models we're developing in my laboratory. So I'd like to start by telling you a little bit about microfluidic devices designed to mimic the human lungs. I think these models are directly relevant to the theme of this webinar series. Okay, so as you probably know, the lung is a very complex organ consisting of functional units called the alveoli. So these are microscopic air sacs deep in the lung that expand and contract during respiration. And these air sacs are covered with pulmonary capillaries to enable gas exchange. And the interface between an alveolus and surrounding capillaries is composed of lung tissue, alveolar epithelial tissue on one side, capillary tissue on the other side, separated by this very, very thin interstitial membrane. So to mimic this interesting structure and also the dynamic environment of the alveolar system, we used microfabrication techniques to build this device called human breathing lung on a chip. So this photo shows the entire device. The device is about the size of a computer memory stick, and it's made of transparent and also biocompatible silicon elastomer called PDMS, polydimethyl siloxane. And if you look at the cross-section of the device, there are these two cell culture chambers separated by this thin porous flexible membrane. So what we can do here is to culture lung cells on one side and capillary cells on the other side to mimic the original structure of the alveolar capillary interface. And what we can also do is to apply cyclic vacuum suction to these side chambers to stretch the tissue layers to mimic breathing motions, cyclic breathing motions in this microfluidic device. So this is a real-time movie of a membrane stretching in our microfluidic device, and the pores shown in this movie are pentagons shown in this movie are pores on the membrane. So this device basically allows us to culture human cells in a very physiological environment. In this case, air on the lung side and blood flow like liquid flow on the capillary side. It eventually allows us to form this living bilayer tissue that looks like the structural unit of the lung. So I'm skipping a lot of details, but we did a very thorough biochemical characterization, morphological characterization to confirm that these tissue layers indeed resemble the original alveolar capillary interface. But so this is interesting, but what's more interesting is that so this model looks like the lung, but also it functions like the lung. So this model allows us to mimic not only tissue-specific, morphology, differentiated morphology and functions, but also more complex organ-level functions, integrated functions that arise from interactions between multiple tissue types. So to demonstrate this idea, I'd like you to think about lung infection as an example. So during a lung infection, for example, these bacterial cells in the lung and the alveolar air sacs stimulate alveolar epithelial cells and also resident immune cells like macrophages to produce and secrete pro-inflammatory cytokines that diffuse through the tissue layers and activate the endothelial cells in the surrounding capillaries. And as you all know, when this happens, the white blood cells, like neutrophils, are circulating in the blood, patrolling the body. Actually, they sense the chemical gradient and also activation of endothelial cells. So what they do is they stick and they squeeze themselves through the capillary tissue into the alveolar air sacs. And once inside the air sacs, they actually are guided by the chemical gradient, and they migrate, actually crawl directly towards the site of infection. And this is not quite how they crawl, but for just demonstration purposes. And when they get there, they try to resolve the infection by engulfing the bacteria. So this is how our lung fights off infection. And as far as I know, this is one of the most complex integrated organ-level functions that occur in the respiratory system. So the important question here is, can this lung-on-a-chip system do this? So let's take a look. So this movie shows fluorescently tagged primary human neutrophils flowing in the capillary channel of the lung-on-a-chip device. So as you can see here, nothing interesting happens. But when we introduce bacterial cells on the lung surface to mimic pulmonary infection, as you can see, these white blood cells actually stick to the capillary tissue because now the endothelial cells have been activated. And so this is my favorite movie. So this neutrophil sticks and curls around, and at some point it starts wiggling its way through the capillary tissue and one of the pores on the membrane to get across the tissue layers into the air-filled lung compartment. And following this, these white blood cells shown in red here chase the bacteria and phagocytose them to clear and resolve the infection, just like what happens in the living human lung. So the nice thing about this model is that because of the optical transparency of the device we are using, it becomes possible to visualize and analyze the entire process of this very, very complex physiological response in real time at high resolution, which would be very challenging to do using traditional cell culture models or even in animal models. Okay, so I think this model is interesting in the sense that this system allows us to mimic complex physiological functions and responses, and we also did nanotoxicology studies, using this platform. And so I just don't have time to talk about all these data, but if you're interested, please refer to the original science paper we published in 2010. But another really interesting aspect of this is, so using this technology, we can start thinking about modeling complex disease processes in the human body. So this is a new research direction my lab is currently working on, focusing on. So just to give you a quick example, when kidney cancer patients or melanoma patients receive this chemotherapeutic drug called interleukin-2, they often develop a complication known as pulmonary edema. So edema is, as you all know, there's a fancy medical term for swelling, and it's clinically defined as extravasation of intravascular fluid in the alveolar air sacs. And in severe cases of pulmonary edema, as a result of enzymatic reaction between plasma proteins and tissue factors secreted by inflamed cells, you end up getting these blood clots, fibrin clots, within the alveolar air sacs. So very interestingly, when this same drug is administered into the capillary channel of the lung-on-a-chip device, so what happens is, as you can see here, so liquid in the lower capillary channel gradually leaks into the air-filled lung compartment, and this channel becomes completely flooded within 3 to 4 days, which is very similar to what happens in these patients developing IL-2-induced pulmonary edema. We also see very extensive clotting, actually, the fibrin clots, forming on the surface of alveolar epithelial surface cells. And so using the system, we can, mimic these clinically relevant key pathological features of pulmonary edema induced by IL-2 toxicity. And more recently, we have also begun to investigate the adverse health effects of cigarette smoking using this technology. So, as you know, smoking has decreased a lot over the past 2 decades. But in the United States alone, 18% of the population still smokes. So smoking-induced or associated lung injuries and diseases remain a very important public health concern in both the developing and developed countries. So anyway, so for this study, what we're essentially doing is to smoke the cells in our microfluidic device. So we're developing a smoking lung-on-a-chip model for this study to investigate smoking-induced abnormal biological responses that happen in the small areas. So the question is, why small areas, right? So there's now very convincing evidence in the literature that these small-sized bronchioles are a major set of tissue infection, injury, inflammation, and remodeling, and they're thought to play a very important role in the pathogenesis and exacerbation of smoking-associated lung injuries and diseases, but not much is known about what happens in this distal part of the lung that is very hard to access. So just to tell you a little bit about the anatomy or structure, the small area tissue consists of this columnar epithelium containing these ciliated cells and Clara cells or club cells, and this epithelium is supported by this stromal tissue containing cells like fibroblasts, vasculature, and macrophages. So to mimic this three-dimensional structure, postdoc in my lab, Dr. Mark Mandrinos, has created a small area on a chip device, which is shown here. So this device has 5 layers. And first of all, to mimic this stroma, what we do is we fill this middle chamber with ECM hydrogel along with the human lung fibroblasts, primary cells, and also sometimes the macrophages. And we also develop a special surface coating technique that allows us to very firmly anchor this ECM hydrogel to this polymeric surface to minimize and prevent gel contraction induced by cell-generated contractile forces. And this layer is then sandwiched between these 2 chambers, 2 channels lined with the primary human small area epithelial cells and primary human microvascular endothelial cells. So using this platform, we can form this highly viable and fully differentiated primary human small area epithelium. And this image also shows a very nice, a stable and physiological stromal tissue that we were able to form in this device. And just for your information, the green shows a cell-deposited fibronectin. We use type 1 collagen gel. So we have evidence that these cells feel comfortable sitting in this environment, and they actually actively remodel their surrounding matrices in the long-term culture in our microfluidic device. So this is how we, culture these cells. But for this particular project, the more important question to be asking is, how do we smoke? So this is how we do it. So this is the smoking setup that Mark has developed. And so we have a smoking machine that generates cigarette smoke in a very controllable fashion. And the smoke is humidified and diluted in this mixing flask to mimic physiological concentrations in the distal part of the lung. And then the diluted sample is delivered into the lung on a chip lined with the cells. So using this platform, we are studying various biological responses to cigarette smoke, and we just don't have time to go into all the details here. So I'll just give you very, 2 quick examples. So it's been pretty well established, as you probably know. Cigarette smoking disrupts proteostasis and significantly increases protein misfolding or unfolding. So when this happens, ER, endoplasmic reticulum, which is a cell organelle responsible for fixing these protein molecules, gets stressed, and that leads to something called UPR, unfolded protein response. So this is a very early response that happens very quickly. So what we are showing here is, as a result of smoking for a couple of hours, these two UPR markers, transcription factors, get upregulated significantly, as you can see in these micrographs. And in the longer-term experiments, what we see is we also found out that the cigarette smoke induces very significant remodeling of the stromal tissue in this model. So this is what the stromal tissue looks like. Just to remind you, green shows a cell-deposited extracellular matrix protein, like a fibronectin. But after smoking for a couple of days, we start seeing these, very extensive ECM deposition within the stromal tissue. We also see increased cell proliferation, and more interestingly, we see two populations. These fibroblasts that kind of retain their typical spindly morphology, but we also start seeing these polygonal cells and very extensive selective pericellular deposition around these polygonal cells. So this is this heterogeneity is an emerging concept in fibrosis, but it was very interesting to be able to see this in our in vitro model. Anyways, so these are some of the examples of biological readouts and responses that we can measure and analyze in this smoking long-on-a-chip device. Okay, so this, I think, the smoking or human long-on-a-chip technology is interesting in the sense that it allows us to mimic both the complex physiological and pathophysiological processes that happen in the respiratory system. So I think, this is very exciting and it represents a major advance from traditional cell culture models. But what's more interesting to think about is, so we can, potentially apply exactly the same biomimetic the ocular system from the external environment, and it we use 3D printing technology to make these 3D dome-shaped cell culture scaffolds with the same curvature as the human cornea. And to mimic the tissue layers, what we do is, first thing we do is we inject ECM hydrogel precursor solution along with primary human keratocytes into the cavities within the scaffold to kind of replicate this stromal layer. And we also developed the 3D cell patterning technique that allows us to form this physiological concentric tissue pattern with corneal cells at the center, surrounded by the conjunctival cells. And for culture, we seed the device, grow them to confluent monolayers, and then we expose them to air to induce a differentiation, which is very similar to lung cell culture. And as a result of differentiation, these corneal cells that start off as a single layer become stratified, forming multiple layers. They form tight junctions, upregulate epithelial markers, and we also confirm that the differentiation of conjunctival cells into goblet cells. And then we combine this with this electromechanically actuated soft hydrogel eyelid layer to mimic blinking motions. So as you can see here, we inject artificial tear fluid into the channel. It gets secreted and picked up by this sliding eyelid and then gets spread on the surface. So here we're showing that even after repeated cycles of actuation, blinking actuation cells remain adherent and viable on the scaffold surface. But the reason why this is important is because this is the mechanism that allows us to form physiological tear film on the surface of the scaffold. So to validate this, we use this instrument called OCT. So this is a clinically used instrument that allows ophthalmologists to measure tear fluid in human patients. So what we did was to measure the thickness of tear fluid layer in our device, and it was measured to be about around 15 microns. And the physiological tear film thickness ranges from 5 up to 20 microns. So we were able to show that this device allows us to form a physiological tear fluid. So I guess I'm running out of time, but so I'll just very quickly mention this. And so we're actually leveraging this technology to develop a platform for screening acute toxicity of chemicals. And also we're using this platform to model complex ocular diseases like DED. So I'll just skip this in the interest of time. And we're also doing a lot of work in the area of reproductive biology and medicine. So this is an area that I think has traditionally suffered from a lack of human relevant predictive models. And so we're very excited about the possibility of leveraging this technology to contribute to research activities in this area. So for this, we're developing reproductive organ on a chip models, and recently we published a paper about papers about placenta on a chip. And we're also developing cervix on a chip. And so if you're interested, please refer to these papers here. Okay, just to quickly wrap up, we are developing these microengineered physiological cell culture devices that allow us to mimic the essential structure and function of the functional units of human organs for a variety of applications. And we think that these models have great potential for a variety of applications. Just to quickly tell the impact that our work has made. So, for example, the human breathing law on-a-chip technology actually had a major impact, and it changed the way regulatory agencies and funding agencies thought about in vitro testing, and became one of the most important reasons why they started new funding programs. And President Obama, made an announcement 5 years ago, highlighting major plans to support research activities that are focused on developing these microphysiological systems for screening for more efficacious and safe drugs in preclinical trials. So we feel very good about that. But more importantly, we as bioengineers, biologists, or medical scientists, working at the interface between biology, engineering, and medicine will have a lot to offer in this very exciting new area. So just a quick mention, we also started a company based in Boston. It's called Emulate. They're translating and commercializing this organ-on-a-chip technology in partnership with the pharmas and regulatory agencies like FDA. And I also gave a TEDx talk last year to talk very specifically about what this technology has to offer in terms of improving the current drug discovery pipeline. And we recently also wrote a review on a similar topic for Nature Reviews Drug Discovery, in case you're interested. Okay, with that, I'd like to end, and I'd like to thank my group and also funding sources. And with that, I'd like to end and be happy to take any questions you might have. Thank you.
Okay, thanks very much, Dr. Ho and Dr. Barabay as well. Certainly some really exciting work going on in both of your laboratories. We do have a few questions let's see, we'll start, actually, this is for Dr. Ho. For the lung on a chip, can we use modeling and or reverse dosimetry to predict external doses that will cause lung injury observed in this in vitro system for specific chemical toxicants?
So what were external factors? I think I missed the first part. Can you repeat the question? Sorry.
Sure. Can we use modeling and or reverse dosimetry to predict external doses that will cause injury observed?
Yes, actually. Challenging task, and so in our case, in our experiments, our doses were based on the values we found in the literature. But for determining optimal doses for for injuries or for treatment, I think what we would have to do is to kind of leverage this technology in conjunction with the the computer simulation models, like modeling approaches like PK to figure out, the resident times, for drug compounds in each organ compartment, and also there are many factors we should consider to determine, optimal doses for treatment, or to find out, the toxicity, caused by, overdosing the drug. So, so we haven't actually tried that in my lab, but other groups.
You may.
Yep. If you look at on the slides, there's Prithik et al. That's for the 2011 paper about the microlung model. But I'm sure, as Dr. Hu knows, you get stymied with the IP, and it's taken for a long time for the metabolung. We haven't been able to publish that yet. We've just secured, the patent and everything. So the university is really wary about me writing anything. So I have to, just do little cursory review papers and things like that. But Zoe's paper, the 2011, you can read that and you'll be able to build the model yourself. Or you can we can license the technology as well.
Sure. Okay, great. Thanks very much. Well, that concludes the questions that have come in via the chat box. So with that being said, I'd like to again thank Dr. Barabay and Dr. Huff for their participation today. This was the 6th and final webinar in a series on alternative approaches for acute inhalation toxicity to address global regulatory and non-regulatory data requirements. We had over 500 registrants for this series, so we're really pleased with the interest and the participation in this series. And certainly, as you can see on the screen, there are hyperlinks where you can find more information on this series in total, as well as on the PISC website, access to the recordings of each of the webinars. So with that being said, thank you very much and have a nice day.
Thank you.
Thank you.
Goodbye.
Thanks.
Bye.